to continue with the biomolecules. Previous lecture, I started with enzymes. Now let us continue with the enzymes. The rest of it, the factors, the different cofactors, which we will be talking about today. Now, what are the different factors that affect the enzyme action? Temperature is one major factor which affects the how an enzyme will work. Now, all enzymes, they need an optimum temperature to show its maximum efficiency. If we plot a graph showing the enzyme activity, so here if it is the substrate, this is the enzyme which is working. Now the enzyme starts working with gets attached with the substrate, and then it starts working. Its activity is maximum activity. That means the reaction actually is possible only when it reaches this optimum temperature. So what is the optimum temperature? That is 40 degrees. When it reaches 40 degrees, then only we see the activity of the enzyme. Till this, when it was 10 degrees, 20 degrees, 30 degrees, it was not showing any activity. So when it reaches to 40 degrees, then it shows maximum activity. And again, if the temperature falls in the system, the enzyme activity will again decrease. So we can say that enzymes can remain active, or they show maximum activity only at an optimum temperature. Likewise, pH. For an enzyme to remain active, it needs an optimum pH. The optimum, when it gets that optimum pH, now that pH might vary. In this particular example, the pH has been taken as 8. That means this particular enzyme can remain very active only when it gets a pH value of 8. A basic, slightly, that means basic. Slightly basic medium when it gets, then it will show maximum activity. If the medium is acidic, the enzyme will not react or not help in the reaction, or if it becomes highly alkaline, even under that condition it will not show its activity. So as like temperature, pH also, the enzymes can work only at an optimum pH. The third factor, substrate concentration. Now we might be having 10 enzymes, but we have a number of the substrates. Now what happens? The number of substrates is more. Now what will happen? The reaction rate will increase till the time all the enzymes are occupied by the substrate. Once all the enzymes are occupied or attached with the substrate, then what will happen? All of them had got occupied, and then at that point it will reach the maximum efficiency level till the products are released. Once the product is released, the enzymes will be free to bind with more of substrates. So the substrate, even if we increase the substrate concentration, it does not mean the reaction rate will go on increasing. It will increase only till the time the enzymes reach the saturation point. Now coming to inhibitors. This is also a factor, inhibitor. That means to stop, to inhibit. Now, there are some molecules or some compounds which resembles the substrates. Here in this particular figure, this is the enzyme. The gray part is the enzyme with the active sites given here, and the substrate is this yellow one. Now, there can be some other compounds or molecules which resemble partially with the substrate. Structure resembles partially with the substrate. As you can see, this lower part resembles both of them, but actually this green is not the substrate. And it so happens, if their number is more, they are more likely to attach first upon the enzymes, though it is not meant for this enzyme. So what happens? It gets attached, not leaving any space or any scope for the actual substrate to come and bind. Consequently, no reaction. Because the active site is blocked, substrate cannot bind, and there is no reaction. So we will not get the product. So thereby what is happening? It is inhibiting. It is stopping the enzyme from showing its from showing its reaction or function. Such an inhibition is called as competitive inhibition. Why competitive? Because these two molecules are competing to bind upon the active site. If the substrate can bind, well and good, reaction will take place. But if the number of the inhibitor is more, the inhibitor will bind. Let us try to understand with an example. Now we have an example here, malonate and succinate dehydrogenase. These two are compounds. Now malonate is a competitive inhibitor of succinate dehydrogenase. Has a structure that is similar to succinate. Inhibition is, now what happens here, malonate and succinate, they have the same type of structures. So what happens now, actually the enzyme, this is the enzyme given here, the enzyme should get attached to the succinate. It should get attached to succinate, form an enzyme substrate complex, and then the product will be released, which is fumarate, fumaric acid. This is succinic acid, fumaric acid. But as because succinic acid and malonic acid, malonate, both of them have similar structures. So what happens, in place of succinate, malonate goes and binds. Blocks it, no reaction. Because succinate does not have any space to bind with the enzyme. So consequently, no reaction or no product. So here, malonate competes with succinate dehydrogenase, the actual enzyme, to bind and block the enzyme active site. But of course, this can be reversed by adding more of succinic acid. If we increase the amount of succinic acid, then the competition will be there, and obviously the number which is more, chances are more that it will win the competition. Now we have to classify the enzymes. Now classification of enzymes is quite simple. It is dependent upon what reaction it helps in carrying out. If it is helping in carrying out oxidation reduction reactions, then we will call them as oxidoreductases. Example, dehydrogenase oxidases. If it is helping to transfer groups, functional group from one to the other, then we will call it as transaminase kinases. Example, this is example. Then hydrolases, if it carries out hydrolysis of a substrate like digestive enzymes, esterases. Okay, so then hydrolysis system is removal of water. So such enzymes will be called as hydrolases. Isomerases, if they are carrying out isomerization reaction, that is they are changing the position of molecular structure. Chemically they remain the same molecular structure. Examples are phosphohexoisomerase, fumarase. Lysis, when they remove a group, break down the compound, then we will call them as lysases. And if they're helping joining together of compounds, we will call them as ligases. So these are the 6 groups of enzymes which have been named according to the type of reaction it carries out. Examples are given here. Now, cofactor. Now, when we are learning about enzymes, now this is one picture of an enzyme. The whole of the enzyme is referred to as holoenzyme. Holoenzyme is always made up of 2 parts. Apoenzyme, the pure protein part, which is attached with one additional part which we call as coenzyme. Coenzyme and apoenzyme together will help in forming the actual catalytic site or active site, which is referred to as the cofactor. Coenzyme is a type of cofactor. In fact, it is actually the cofactor. So a holoenzyme is formed by one apoenzyme and a cofactor. Here I have it. So here apoenzyme and this cofactor binds with it, completes the enzyme structure. This will now bind with the substrate, or is capable of binding with the substrate to carry out the reaction. Now the thing is here, this cofactor may be permanently bound upon the apoenzyme, or it may be temporarily bound. Like here you can see, this is the apoenzyme. So it becomes active when the cofactor comes and binds with it. Then the active site will appear. Thereby, what will happen? The substrate can come and bind with it. Substrate can bind with it. With what? With the holoenzyme. So the holoenzyme, what you have to remember is, the holoenzyme is formed only when apoenzyme, the protein part, binds or combines with the cofactor. Now, there are types of cofactors depending upon their binding ability. Now, these are the different types. Enzyme cofactors. Some enzymes require cofactors to be active. Okay, here enzyme is protein only. Example, lysozyme. Cofactors are a non-protein component of enzyme, which may be organic molecules or inorganic molecules. Now, if the cofactor is permanently or tightly bound upon the apoenzyme, tightly bound, then we call it as prosthetic group. Example, flavoprotein. We call it as prosthetic group. If it is permanently bound, tightly bound upon the enzyme, and if it is temporarily associated, that means it will bind only when required. If not, it will remain free, thereby keep the enzyme inactive. And whenever it has to get active, it has to attach with the substrate, then the coenzyme will come and bind with it. This is temporary attachment. Here the example is usually coenzymes. Coenzymes are usually temporarily associated. So, which can be again some vitamins like niacin, metallic ions like zinc, magnesium can also behave as the coenzymes. So these are the different types of cofactors which are responsible for the functioning of enzymes. So that is all for this chapter. So I hope you could follow all the lectures, the series from the beginning till the enzymes. Thank you.